Okay, now we're going to take a couple of minutes and look at the actual problems that happen when our Reduviety, when our kissing bugs get involved with our animals. So a major disease that is spread by Reduviety is called Chagas disease. It's caused by a plasmodium known as Trypanosoma cruzi. So this is a protozoan that is found in the hindgut of true bugs. These protozoans are imbibed by the bug during feeding, so they come out of the blood of animals and humans, and they multiply in the midgut into huge numbers that line the walls of the insect's rectum. They'll pass out of the bug via fecal matter. So you remember when we talked about the Reduviety, they feed on you, then right before they leave, they poop on you. This is when this protozoan gets out of that rectum during that fecal matter expulsion, shall we say. Okay, so they pass out of the bug via the feces, and they infect a new vertebrate host when that infected feces is rubbed into a bite or into a break in the skin or into the eyes. So think about this, right? You've got this insect. It's feeding on you or your animal. This feeding is painless for the most part until that insect leaves and stops pumping in that anesthetic. It poops on you right before it leaves, and then you scratch that really itchy bite. You're scratching that fecal matter in there and potentially this protozoan. So it may also be passed when the feces comes into contact with other mucosal membranes, like your eyes or your mouth or something of that nature. So like with an animal, if they lick that wound, they end up licking it up. Or if you rub your eyes after getting it on your fingers, that can happen. Once a bug is infected with trypanosoma, it's infected for life. So it can continue to spread this disease from host to host for the rest of that 3-year lifespan. Chagas disease is distributed throughout the Americas, and there's approximately 12 million human cases of the disease. We get about 21,000 deaths from Chagas each year from this protozoan, and there are very limited treatment options when it comes to this. Common symptoms of Chagas disease are things like localized swelling at the infection site. You get lymph node enlargements. You get tissue changes in the presence of the protozoa. These protozoa are going to be in your bloodstream. You're going to have fever, an enlarged liver, an enlarged spleen, and skin rashes. Complications may result in the death of the host. Now, we are in a veterinary entomology class, though, so let's talk about how this Trypanosoma species actually affects our animals. The major issue with this hemiptera in the veterinary realm is this trypanosoma that it spreads to animals. And the one you are going to likely run into, we call canine trypanosomiasis. It's basically the canine version of Chagas disease. So canine trypanosomiasis comes in 2 major forms. First is called American trypanosomiasis, which is caused by Trypanosoma cruzi. It's the same agent that causes Chagas, which we just talked about. But there is another agent which is grouped into this canine trypanosomiasis. This is called African trypanosomiasis. It's caused by Trypanosomiasis evensi, which causes sleeping sickness in humans. So slightly different disease in humans, but we lump it into this trypanosomiasis in canines or in dogs. So trypanosomiasis was originally an enzootic disease, meaning that it circulated just in wild animals like mammals and birds, and they served as reservoirs for many years. It was this domestication of animals and humans expanding into traditionally wild areas that allowed this trypanosomiasis to jump to humans and our domesticated pals. So our dogs, our cats, that sort of thing. Dogs are the main domestic animal that is involved in the transmission and the maintenance cycles of these parasites. So that's why we're going to talk about this, and this is where you're most likely going to run into this in your veterinary life. Dogs can develop pathological symptoms that are very similar to those found in humans. So actually, we use dogs as experimental models for Chagas disease. So the prevalence of T. cruzii in dogs, we can use that to figure out what areas humans are going to start getting Chagas, because those areas where there's a lot of T. cruzii present, dogs are going to get it first, then it's going to spread to humans. Now, Dogs get both T. krusei and T. evansii primarily by ingesting infected triatomines. So they ingest the actual bug. You've seen dogs before, right? They're going to feed on whatever is out there. They're going to bite it. They're going to eat it. But they can also get it through feeding on the organs of animals that were killed by trypanosomiasis. Maybe infected fresh carcasses, infected rodents or other wild animals. If they go off hunting those animals that have this trypanosomiasis, they can get it that way. They can also get food that has been fouled with kissing bug fecal matter. If they feed on, say, just kibble that kissing bugs have pooped all over, that's how they can get this as well. So there's a lot of different ways that trypanosomiasis can get in there, but it's most often contracted by dogs actually just chewing on an infected bug. So dogs infected with T. krusei, let's start there, can develop both acute and chronic trypanosomiasis. The acute phase, this means it happens immediately. So this happens at least nearly immediately after infection. It can be observed most easily in very young dogs, somewhere between 5 and 6 months of age. The affected animal is going to develop extensive lesions on the myocardium or on the heart. You can see a picture of those there. They also have these lesions in the central nervous system. Then they will be off feed. So they'll develop anorexia. This is inability to feed or this lack of hunger. They're going to get swollen lymph nodes. You can see that poor puppy dog there, those big old swollen lymph nodes. They'll present with diarrhea, with inflammation of the heart muscle, and then sudden death due to irregular heartbeat. Now, in older dogs, it's a little harder to diagnose this acute phase. You get the same basic sort of thing, but it's not really obvious. It's masked a lot of the time. So you may get a dog that's a little off feed or something of that nature, but you can't really tell any major symptoms. Now, the chronic form, chronic means it lasts longer and it happens later after initial infection. So the chronic form in dogs will happen between 8 and 36 months after initial infection. It's characterized by irregular heartbeat, by dilated heart ventricles. That's that bottom picture right there. These dilated heart ventricles that start on the right side and then move bilaterally. In general, an older dog is better protected from T. krusei than younger dogs. They get fewer severe symptoms. Weird, I know, that doesn't really happen in humans, but older dogs tend to have fewer symptoms, and the symptoms are less severe than younger dogs. Now, dogs that are infected with this other group, Trypanosoma evansii, this presents a bit differently than T. krusei. Naturally infected dogs will show intermittent fever. They'll have progressive anemia, so that lack of red blood cells. They'll lose their appetite. They'll have a weakness and a wasting away of muscles. They'll have water retention in the tissues of the head, in their face, their throat, and their limbs, so that they'll start swelling up in their face, head, throat, and limbs. They'll get swollen lymph nodes again. They could present with paralysis in the rear limbs. They'll have this inability to walk. This can move into seizures and eventually corneal opacity. So that's what's happening with this poor dog down here, and a close-up of the eyes there. So it goes into the cornea and turns it opaque. So if you think about it, though, a lot of these symptoms are pretty generic in both of these species of trypanosomiasis. It's really difficult to simply diagnose this type of trypanosomiasis based on these generic symptoms. So we have to run very specific lab tests in order to determine the type of trypanosomiasis. I'm not going to edit that out, screw it, that we're dealing with. Now, because the treatment of this disease depends on the species of trypanosome that you're dealing with, you need to figure it out. Now, T. evansii, this is much easier to deal with. So this African trypanosomiasis, we have drugs that are proven effective in animals. So there's a treatment schedule, and you can usually cure it. T. krusei, much, much harder. We do not have any drugs that will fully cure the disease. There are a few things that can help, but the side effects are pretty nasty of those different drugs, and you have to really consider what is going on with your animal and if it's worth it to give them those drugs. So the best way to deal with this disease in animals is to completely prevent it. You want your dogs to be kept away from kissing bugs. You don't want them to be bit in the first place. You don't want any of that area to be fouled by the kissing bug fecal matter. Now, There are a few other Trypanosoma species out there that will infect animals, but these two are interesting. So Trypanosoma wrangeli, this is found in the same areas as T. krusei, same areas as Chagas disease, but wrangeli is not pathogenic. So animals will get it, and you can see a blood smear here on the left with wrangeli in the blood. However, the animals that get it do not have symptoms. It's just there's protozoan in the blood, and they're fine. How amazing is that? So you can have an infection with this with no adverse effects at all. Interestingly, T. wrangeli shares a lot of the same proteins on its membrane as T. krusei. So what this means is that if an animal is infected with T. wrangeli, it's basically training the immune system of that animal to recognize this protozoan. So there's been a lot of research as of late of using T. wrangeli as an inoculant or as a vaccine against T. krusei, and they've had really great results. So we can inject, say, a dog with T. wrangeli, it'll train that immune system, and then if that dog is exposed to T. krusei, to Chagas disease, they're going to fight that Chagas disease off really, really well. So we're testing it as a way to vaccinate both animals and humans against Chagas disease. Trypanosoma cornori, this is also a non-pathogenic parasite. It's transmitted through the fecal matter of kissing bugs, same exact way as everything else. And primarily, it focuses on rats all over the world. So we tend to see this more often in rats. Now, the reason I wanted to mention these two is that they are morphologically nearly identical to T. krusei. So T. krusei is up there in the right, and the other two are here on the left. It is really hard to tell the difference between these two species. So if you just say do a blood smear of an animal and you come up with this plasmodium or this parasite in the blood, they may or may not actually have Chagas disease. They may or may not actually have African trypanosomiasis or American trypanosomiasis. So you don't want to have to treat with some of these really nasty drugs if they have, say, wrangeli or cornori, which are just completely innocuous. So I wanted you to know about them. So a simple blood test, a simple blood smear is not enough to determine if an animal is actually infected with say, canine trypanosomiasis. You have to look for the symptoms. You have to look for this sort of nasty result to see if you need to treat or not. All right, so that is pretty much it when it comes to what is really, really important with the red uveitis and with the kissing bugs. Let me know if you have any questions.